Conflict over spontaneous generation. Spontaneous generation, abiogenesis. From earliest times, people had believed in spontaneous generation, abiogenesis, that living organisms could develop from non-living matter. Even the great Aristotle, 384–322 BC, thought animals could originate from the soil. This view was finally challenged by the Italian physician Francesco Redi, 1626–1697, who proved that gauze placed over a jar containing meat prevented maggots from forming the meat. Similar experiments by others helped discredit the theory for larger organisms. A evidence prosum proposed that microorganisms arose by spontaneous generation, though larger organisms did not. John Needham, 1713–1781, the English priest, in 1745, published experiments purporting the spontaneous generation, abiogenesis, of microorganisms in putrescible fluids. Félix Pouchet, 1859, the French naturalist, claimed to have carried out experiments conclusively proving that microbial growth could occur without air contamination. This claim provoked Louis Pasteur, 1822–1895, to settle the matter once and for all. B. Evidence on spontaneous generation experiment. Lazzaro Spallanzani, 1729–1799, an Italian priest and naturalist, opposed this view and boiled beef broth for an hour, sealed the flasks, and observed no formation of microbes. Franz Schultz, 1815–1873, Theodor Schwann, 1810–1882, Georg Friedrich Schroeder and Theodor von Dusch attempted to counter such arguments. Louis Pasteur, 1822–1895, settled the matter once and for all. In a series of classic experiments, Pasteur proved that all forms of life, even microbes, arose only from their like and not de novo. Pasteur was able to filter microorganisms from the air and concluded that this was the source of contamination. Finally, in 1859, in public controversy with Pouchet, they prepared boiled broth in flasks with long narrow gooseneck tubes that were open to the air. Air could pass, but microorganisms settled in the gooseneck, and no growth developed in any of the flasks. If the necks were broken, growth commenced immediately. Pasteur had not only resolved the controversy by 1861 but also had shown how to keep the solution sterile. Tyndallization. John Tyndall, 1820–1893, John Tyndall, 1820–1893, the English physicist, finally dealt a final blow to spontaneous generation in 1877. He completed the story by proving that dust did not indeed carry germs and that if dust was absent, broth remained sterile if directly exposed to air. He was able to explain satisfactorily the need for prolonged heating to eliminate microbial life from infusions. Heat-stable form and a heat-sensitive form. He exposed infusions to heat for varying time and concluded that bacteria existed in two forms: a heat-stable form and a heat-sensitive form. Heat-stable forms were destroyed either by prolonged or intermittent heating. Intermittent heating, now called tyndallization, killed both forms since the heat-stable forms changed to heat-sensitive forms between periods of heat treatment. This method of tyndallization served to eliminate many of the anomalies reported by the advocates of heterogenesis. 2. Heat-resistant forms as spores. Ferdinand Kohn, 1828–1898, the German botanist, discovered the evidence of heat-resistant forms as spores. Spores as well as vegetative forms were responsible for the appearance of microbial life in inadequately heated infusions.